Okay, so let me introduce you, Herman. Excuse me, Chris Apfel, MD, PhD, MBA, President and CEO of SageMedic Corporation. You co-founded SageMedic with your wife, Dr. Brigitte Apfel, after several of your family members were touched by cancer. Chris is a physician scientist, board member, and an active angel investor in over a dozen early-stage life sciences companies. You are recognized as an international authority on designing, conducting, and publishing clinical trials that have changed medical practice. For example, being the developer of the Apfel score, a standard tool that has changed clinical practice worldwide. You're a California licensed physician and received your MD and doctoral degree in medical sciences from the University of Gießen, Germany, an MBA from Wharton University in Pennsylvania. The title of your talk is going to be, and I think it's here, Functional Precision Medicine, the Future to Overcome Limitations of Genomic Testing. We are really excited to have you here. Thank you so much.
Well, thank you for the nice introduction. It's my pleasure to be here, and thank you for everybody who is joining this presentation. We are engaged in developing an assay that will, as I said, overcome the inherent limitation of genomic testing, and it was born out of a personal experience. As you know, I've done a lot of clinical research myself, but at some point my dad was diagnosed with lung cancer. He refused any therapy because when my mother had ovarian cancer, he realized that even today, cancer treatment is more or less a hit or miss experiment, and the treatment that she got actually made her rather worse than better. So this is a serious issue for me. I'm actually an anesthesiologist and intensivist by training. I still have an adjunct professorship at UCSF in the Department of Epidemiology and Biostatistics because of my expertise in clinical trial design. But at the same time, what I realized is very quickly, we all think that we are doing a good job in precision medicine. And as you will see, it may not be as nicely as we would like to believe. When one of your loved ones is affected, you will often very quickly realize the future is not here now. We are still living in the past. And so I actually stepped out of my academic role. I actually got my MBA from Wharton. I started the company on my own dime. We've raised some capital. We have now developed an assay that we believe can make a much bigger difference in people's outcomes than any drug that's currently in development. And the key to get this to market is clinical trials. So let me start and give you a little bit of background on how we think about it and where we are coming from. I think I mentioned originally that in the media, from the media, we have the impression everything is good. We are making great progress. Cancer death rates are declining. If you will believe this graph, it will soon be zero, and hopefully will not be negative, because that would be a terrible story. But if you really look at the data, cancer death rates aren't declining that much. It's actually declining very little, and most of which are declining is because we have now less smokers. And lung cancer, even today, account for over 25% of cancer deaths. There are actually more women dying of lung cancer than of breast cancer. We are often not aware of that. So, what's the problem? Why is it so hard for us to make a difference in patient outcomes? Well, the secret is in the publications. If you read JAMA Oncology, you find that the study from Marquardt and colleagues from Vinay Prasad's group at OSHU has actually shown that only 5% of patients benefit from genome-driven target therapy. We all think if we do genomic testing and big data, we'll have the answer. The challenge is that more often than not, patients do not have those genomic mutations. This is the NCI MATCH trial, where the vision was to really expand it and get it beyond get more utilization based on mutation rather than on location. And they have done an amazing job. Now there are probably 20% have been able to match for one drug, and it will continue to rise, but it will always remain limited to the minority of patients. So this is the interim results from the NCI match trial. We can probably now match 1 out of 4 patients with a targeted therapy. But that's it. What about the remaining? What about the remaining 75% of patients? They get traditional cytotoxic therapy, and that's often not good. Now, in terms of genomic testing, we have also taken the next step. We are now doing liquid biopsies. That's something that has raised significant investment interest and has also raised the market capitalization of large companies that are in these liquid biopsy space. Much nicer than having to get a biopsy, a cancer biopsy from a patient. That's not fun. And the rationale is some of the genes will actually get into the bloodstream. And so that would probably be a better approach to personalized cancer treatment. Well, probably not. Because if you look here at the data, so this is a validation study, the results usually don't match with the gold standard of tissue biopsies. The red ones are here only detected, these are 28 patients. The red ones are only detected in the blood. The blue ones are only detected in the tissue, and only the green ones are really concordant. That's really disconcerting. A similar study from Kuderer and colleagues, also published in JAMA Oncology, had a similar result. The results from Foundation Medicine's mutations did not match the mutations that Guardant 360 has detected in the liquid biopsies. And the recommended drugs are, as a result, very different. So depending on to which physician you go, whether he or she prefers the tissue biopsy or just skip it and let's just do some liquid biopsies, you would get a very different treatment. That's very disconcerting. Now, the argument is, well, if you're in a late stage and you take a tissue biopsy, there may be other mutations that are not detected with just having a selection bias on a specific biopsy, and therefore a liquid biopsy may be more representative. Well, if that were the case, so that's the argument on the tumor heterogeneity, then you would expect that liquid biopsy technologies should at least be concordant. This is a comparison between both CLIA certified commercially available tests that are commercially quite successful, Guardant 360 and Plasma Select. The red one with a P was only detected here, the mutation in Plasma Select, with a G only in Guardant 360, and only the green ones are concordant. Now, this is currently the status, and that's the reason why very few pathologists and usually oncologists are very skeptical regarding liquid biopsies. But it's still a big business. And why? Because very often, if you don't find a genomic mutation, you have this as an option, as a second opinion, second try to find something. Perhaps we can apply a targeted cytotoxic therapy. But both oncologists and pathologists do actually know that the gold standard is actually the tissue biopsy. So this is actually the challenge that we have when we think about the traditional approach with the cytotoxic therapy. If we had 100 patients, on average, it differs between tumors. Only 30% of patients have an objective tumor response. About 50% of patients have some response with some interim betterness, and there are 10 to 20% who don't respond at all. And many of them actually get resistant, which is why the therapy with a traditional cytotoxic therapy is really not satisfying. There are huge side effects, especially when it's given for a long run, and yet very rarely there is really a cure. But instead, what we have done in the past, we have actually focused on the minority of patients who then have a higher proportion of responding to the targeted therapy. and that has benefited patients. On average, the duration of a tumor response is about 2.5 years for patients who receive targeted therapies, and there are more patients who are responding to targeted therapies when they have a mutation. It's still not 100%, but still it's better than nothing. The challenge is it doesn't help the majority of patients. We believe that with our genome-free precision medicine platform, our SageDirect test, we can actually help those patients. We can actually significantly improve their chance of a tumor response by identifying ahead of time which therapy, if any, is likely to be effective. And so what I'm proposing here is that in the future, we will realize that we need to embrace functional profiling to make sure we have an approach that can help all patients, not only the minority of patients where we find the needle in the haystack, the specific mutations. We actually need to know which drug is going to work in a given patient, no matter whether there is a genomic alteration or not, and we need to know whether it's going to work on that tissue. And the challenge with a traditional diagnostic approach is that we are actually using this on genomic tissue, on dead tissue samples. We're looking at the DNA, which is only half of the story. When there is a live biopsy taken, this biopsy is actually thrown into formalin, so the tumor is dead. And then we try to extract molecules to understand which one is likely to work had we kept this tumor alive. We are shortcutting this. There is much more going on in a cancer. It's the gene expression, the epigenomics, the proteomics, the metabolomics, cell-cell interactions, the extracellular matrix. There is a lot going on for a cell, which is why it's so hard to predict which patient will respond to a therapy just based on the DNA alone. So therefore, taking a live tissue is one approach that has been done before, that is much more holistic because it includes everything. This is a clonogenic assay that was developed by Anne Hamburger, a woman at the time, published first in the New England Journal of Medicine by Sidney Salmon, who also unfortunately passed away from pancreatic cancer in the last couple of years. But they have developed a test where they actually were growing tissues in vitro and then having drugs on it and a control group. And if those drugs could inhibit growth, and I make a long story short, they were able to show with 80%, roughly 80% of accuracy, whether a drug or a regimen is likely to work or not. That's an impressive result that was studied extensively. Dr. Daniel Van Hoff has been very active in this group, has published in the Journal of National Cancer Institute 1990 these data on over 2,000 patients. And yet, nobody's using it. Yet most oncologists, if you approach them, are under the impression that, oh, if you take a patient out of the patient's body, if you take a tumor out of the patient's body, it can't work because it will be different. No, it's actually interesting that even in 1990, the data were already out that if you have an in vitro result that responds to a therapy, that it will also be a patient response in the patient's tumor. The reason here why this didn't make it into clinical practice was that growing the tumor in general was so difficult. So only 1 out of 3 patients we had those tumors could grow in the dish so that we had a result that we could act on. If we had the results, it was highly predictive. But it usually took too long. It usually took several months to grow the tumors in vitro. And in the majority of cases, they didn't grow at all. So therefore, a different assay was developed, the extreme drug resistance assay, where they actually use very high doses that would normally not be clinically achievable. And if the tissue stayed alive, nevertheless, there could at least be made a prediction on this being predictive for what is the patient's tumor is resistant to. Long story short, clonogenic assays and now organoid models that are in academia, they often take too long, weeks to months to grow, and often they didn't grow at all. The cell death assays and the extreme drug resistance assays, they often had their own limitations. For example, they were very specific. They could predict to what the patient was resistant to, but were not very good in predicting what the patient would react to. So we have developed, therefore, a Sage Direct test where we overcome these limitations. We have asked oncologists whether that's something they would actually order. If we can increase and if we can show with clinical trials that there is an improvement in patient response by about 20% and an improve of 1 year survival by 10%, and basically every oncologist would say, yes, that would be an assay we would order if there were 2 double-blind randomized trials that demonstrate that the assay itself could help an oncologist to improve patient outcomes. That's actually usually not done. Usually in the diagnostic space, to demonstrate that it has a clinical impact is a very, very high bar, but it needs to be done. So what we have done is we have developed a test where we can, based on a patient's core needle biopsy, we can create hundreds of 3D microtumors, treat them with an array of drugs, and then measure the tumor response with a result back for the oncologist within 1 week. This is a big deal. And we can do this basically in every viable tissue we get, a normal tissue biopsy, as long as it is shipped alive, cool, in a cooled chain to our lab overnight. We believe this can actually double the patient's chance of a tumor response and significantly improve patient outcomes while reducing costs. That's the reason why I left academia. We have developed this technology. We can maximize the yield of fragments that have the original tumor microenvironment and the cell-cell interactions. We then can create those 3D microtumors within less than 24 hours, and we can then observe with confocal microscopy how they are responding to the various therapies so that we can see how the tumors are changing as a result of the treatment. If a treatment is effective, we can then quantify this treatment and translate this to a score from 0 to 100. So this here gives you examples of 7 ovarian cancer patients where we have exposed those microtumors to the range of different drugs, both traditional cytotoxic therapies as well as targeted therapies. And so we have now developed a method that can be used to predict the response to patients. That said, it will also require calibration and clinical validation, which is what we are currently working on. And this is actually also a platform that could be used for drug development. This is actually the key why we can make pharmaceutical companies select the most effective drug more efficiently. As you may know, only 10% of drugs that enter phase 1 will ultimately get FDA approved. Many of them fail because of lack of efficacy. And the traditional approach would be to use a patient-derived xenograft, grow it in mice, and see, once it's been grown in mice, whether those drugs are working and which drug would be best. We can do this much faster, cheaper, and better because we have a result in 1 week and at a much lower cost and have, therefore, a much more consistent result. We have run this by key opinion leaders who are impressed with what we are doing. And our next step for the clinical development is to start off with a tissue collection and tissue donation study, as well as with a non-interventional correlation study to calibrate the assay, because what we see in the lab needs to be calibrated with the patient outcomes, in this case, tumor response. And that's then the foundation for subsequent clinical trials that can then lead to definitive randomized control trials, in which case we may also be talking with the FDA to get a claim on improved outcomes. So in summary, what we do at Sage is very personal to me. We have actually invested significant amounts of our own private savings. We have raised some capital through angel investors. We are now raising our Series A, starting to raise that soon in order to support our clinical trials. And this is important because genome-driven targeted therapies has limitations, and because we know that the only way to really understand which drug is likely to work is trying it on the tumor itself. And that's what the Sage Direct test is actually doing. And that's the reason why we are so excited about this solution, because we know that this can help millions of cancer patients. So I would like to thank you for your patience with me, for sitting in here, and I would like to open the floor for discussions.
Well, thank you again, Dr. Apfel, for your presentation. And I know there's someone in the audience, Dr. Harumi. If you're there, I know you specialize in precision medicine for cancer research with your group, previously with Notable Labs, and now I think with Tyra Biosciences. So if you're still in the room, if there's any comments you want to personally add, please let us know. Otherwise, I'll give it up to Dr. Apfel.
Any thoughts? Any questions?
I'm happy to break the ice here. First of all, let me tell you that I'm very impressed with what you have presented today. I've been following up all these liquid biopsy companies. I even one that is not so much in the radar screen of many people, like Freenome, besides Current Health and Grail and Foundation. And I'm aware of those discrepancies that you're mentioning. So I think that's something that still is obviously not relevant enough to stop this fast-moving train, which I'm very, very, very excited about what you mentioned. I'm also currently working as an advisor for Notable Labs. And there are also some similarities in what you're doing there. So at some point, I was one of my roles, chief medical officer with Myriad Genetics. And my question to you is something that you have mentioned, and it's very impressive, your non-interventional clinical trial that you're conducting, and that you're is not really looking into making any changes, although I think on the therapies of these patients, although that's something that is needed at some point. But my question goes more into something that was absolutely critical, and it is also now for all these companies and for you, also when you are thinking about the investment rounds at some point, funding rounds. And this is in regards to Medicare reimbursement. And you know that that's absolutely critical. At the end of the day, someone has to pay for it, and the way to start is with CMS and Medicare, and being able to achieve these kind of LCD, which is the way these contracts are starting to be rolling in. So my question is, how are you dealing with that? Who is going to pay for that? So how are you thinking in terms of presenting yourself to the investment community from a perspective of being available to the public or to the patients through either Medicare or private insurers?
You brought a lot of things to the table here, and let me make 3 responses. I really appreciate it. So first of all, you mentioned the liquid biopsies, and I would like to clarify that liquid biopsies are used for 2 different areas. One is to screen whether the patient might have cancer, and Grail has fantastic data, and there are 2 or 3 other companies, I'm very excited about it, that might be able to help to identify a patient with cancer patients years earlier, before symptoms occur. And I believe that a major quantum leap is about to happen once we are able to detect cancer earlier. So in that regard, I do think that liquid biopsies are a wonderful development and are exceptionally important and useful and will carry significant benefit for patients. It's very different once a patient has been diagnosed with cancer, whether that can actually help to guide therapy. That is a big question mark, and that was what I was alluding to. You also mentioned that you're an advisor of notable labs, and they have actually done a fantastic job in basically also having a phenotypical approach. They also use the patient's live tissue in a way as hematological tumors, either from the blood or from the bone marrow. And with their flow cytometry technologies, they can get a very deep functional profiling insight into what's going on and which drug is likely to work best. So I do think that notable labs can only be congratulated for their efforts and for their accomplishments in what they have built up. And I do think that they fill an important role, an important need. Reimbursement in the clinical pathway, that will definitely be a challenge in the initial phase until we have been able to show both that we can improve patient outcomes, and second, that it is economically feasible. And so I have actually led a health economic analysis for Cadence Pharmaceuticals years ago. I was able to show for a drug that did not have a reimbursement code, interestingly, $500 per patient cost savings to hospitals. And 6 months later, we were acquired for $1.3 billion by Mallinckrodt. So reimbursement is one part, but actually it doesn't have to be in reimbursement. It needs to be economically feasible for at least one party that is actually paying. That's the key answer. But before we get to the reimbursement, the main issue is, how do you get the clinical trials done? Because the reason why liquid biopsies have so good traction is because it's so convenient. Getting a tissue biopsy from a patient, from a cancer patient who is currently sick, is a relatively high bar. And so the conduct of clinical trials, unless we're talking about donations of tissue that is residual tissue after surgery, the conduct of clinical trials and showing the correlation does require a significant effort to get this off the ground. Once that's done, that would be fantastic. But a major barrier to entry is actually to run those clinical trials and make them work so that we can demonstrate that patients can benefit from it.
Excellent response. Thank you so much. I think that makes things very clear for everyone, and I love the topic that you brought up in terms of prevention, which is a very important area, and the fact that some things are not so easy to perform, but you're moving forward and you're getting us to a point where we will be able to really identify those patients that benefit from certain types of therapies. Thank you so much. Great.
And there is, I think, one question from Sandy Imboden from Clinical Maestro for you. She asked, is there a specific window of time to get the tissue sample from the patient and then begin growing the microtumors?
Yes. The specific time is right away. the carpe diem, I would like to say, the opportunity is here now. When a patient gets a biopsy, we have a shipment kit. It needs to go straight into the kit, into the shipment kit, and then will be shipped overnight on a cool chain. It needs to be at the time the biopsy is taken, and so that's an important part. The other part I wanted to clarify is the key is why we have overcome previous limitations of functional precision medicine of clonogenic assays is that in our 3D microtumor approach, we do not need to grow the tumor. We culture the tumor, and therefore we can have a result within a week.
Wonderful, wonderful. Awesome. So I do want to thank you again for presenting, Dr. Apfel. And Dr. Oscar, if you have any final remarks for the audience as we wrap up.
Thank you so much, Sam. I would like to start with you and with your team. I think that you guys have been doing a phenomenal job today organizing all these. I love also the platform that we have been using. Took me a little bit of time to click in the go live, but I think that it has been working perfectly. The speakers, also panelists yesterday, are really phenomenal. So you guys did a very thorough work identifying all them, and that makes easy the job for a chair when you have these kind of tremendous people. And even our last speaker and the fact that we have a couple of things in common makes me extraordinarily excited about this. So looking forward to your next session or meeting, which I think you are calling this West, so I guess there is going to be an East one. And again, congratulations to everyone, and thank you for this opportunity to be here with this extraordinary faculty.
Thank you too, and please reach out. Feel free to reach out, Senor Segurado.
I will. I'm about to send you an email.
Excellent. Fantastical.
Fantastic.
Yeah, I want to thank you both, and especially Dr. Oscar, for chairing the entire day. I know he's had meetings that he's been going back and forth answering emails. I want to thank you, all of our speakers, Dr. Apfel, the panel discussions, those in the audience, all of our sponsors. Indeed, our East Coast meeting will be May 12th, May 13th.
Oh, okay.
Interested in speaking or attending that, you can just reach out to me directly. I can help secure your slot for that. In the meantime, if you want to network with anybody, feel free to message them in the chat box over here in the lounge, visit any of the booths in the lobby. And until then, I wish you all well. Take care. Good afternoon. Good night. Bye-bye.
Thank you. Bye-bye.
Okay. Can you tell me how to get into the lounge then? Or you will just put me there, right? Is that correct?
You can just exit the room. If you hit the go backstage on the right top
okay.
right, and then the lounge is on your left-hand side.
Excellent.
Lounge, yeah. You can just pick a spot there.
Okay. Thank you so much. Bye-bye.
You're welcome.